Hi everyone, I'm Giulia Cantini. I'm a PhD student at the Lab of Computational RNA Biology at Helmholtz Munich. And so here I will talk to you about miRXplain, how we decode microRNA and isomiR binding using transformers. First, a very quick reminder for the audience on what are microRNAs. So they are very small non-coding RNAs, which by binding their target transcript, they impact gene regulation by either degradation or translational inhibition. And they typically occur in nature in what is called the canonical form, but it can also occur as variants called isomiRs. And these variants present at the sequence level some mutations. They can be inside the sequence or at ends as insertions or deletions. And there has been emerging evidence, so like studies that have shown that these isomiRs also are biologically active, but compared to the canonical forms, they are under investigated. So in this project, what we wanted to do was to develop an accurate and interpretable model that can predict microRNA and isomiR target binding, with which we wanted to essentially advance the functional characterization of these isomiRs. And we started for this by collecting and curating 5 different public data sets based on chimeric CLIP. And very briefly, chimeric CLIP, unlike the predecessor CLIP, is a protocol that allows to capture the exact target size of the microRNAs to the mRNA, thanks to the introduction of this ligation step here that essentially generates a read that is made both by the microRNA and the mRNA that can then be aligned by reference genome, and you can recover the mRNA part, and then against annotated microRNAs, which you can actually get the miRNA part. And in this project, we essentially introduced an additional step, so an alignment against the microRNA hairpin, with which it was possible then to annotate also the variants. And if you want to know more about CLIP and CLIP-L, there's also a poster that is presented by my collaborator Ranjan that talk about how we benchmark different deep learning methods on this kind of data. But coming back to us, for the data set that we collected, the first thing that we noticed was like a positional sequence bias present at the level of the target sequences. And this bias is essentially an overrepresentation of specific nucleotides at a given fixed position. And we saw that it could be found only on some data sets over the 5. And for this bias, we have corrected before generating the final set of positives that we then need to train our classifier model. While to build the negative set, we followed a procedure that is based on essentially random sampling from unbound region of the 3' UTR of the positive target site, after which we apply some constraints that essentially make sure that this is a good negative. So the first is a constraint based on the GC content of the target site, and the second is based on the computation of hybridization energy with RNA cofold, where we essentially select the final negative as being the one that could yield the interaction that is the least probable. So it's very far from the positive. And this is miRXplain. It's essentially a transformer model, takes in 2 different branches, and the mRNA sequence and the target sequence separately, pass them through embedding and positional encoding, and then at its core it has 2 transformer encoders with an hybrid attention mechanism. So we have 1 layer of self-attention that alternates with 1 layer of cross-attention, with which we kind of aim to capture basically the intradependencies of the sequences and also the interdependencies between the microRNA and the target sequence. And after that, we have some ways to fuse together the projected embeddings, and the final interaction map is used inside the sigmoid to perform the classification. And this architecture was benchmarked against previously published models, among which a baseline convolutional neural networks and some hybrid models like meter and meso, that are essentially convolutions and recurrent neural networks, but also graph networks like GraphTar and another transformer like TechMyTarget. And the results that I will show you in the next slide, they are based on a 10-fold cross-validation setup. And what we saw is that actually miRXplain outperforms all the other models that we benchmarked in all the isomiR categories as we define them. And you can see it here when we compare in the plots showing AUPRC and AUROC. miRXplain is the last box here. And this, so being more performant, but also more efficient, as having way less model parameters than the competitor. And also, interpreting miRXplain is possible if we extract the scores, so the weights, from its attention layer. So for example, here, if you look at this miR21 and 3 of its isomiRs, you can see represented a positional shift, essentially, that corresponds to the seed region of the microRNA on the respective 2D attention maps. And then if we compact the attention maps into attention vectors, so performing a column-wise average, we can easily highlight different regions along the microRNA sequence. So for example, if you look at the line plot for the canonical microRNAs on the top right side, we have highlighted the seed region in red, the bulge, and the 3' compensatory region in green. And here we can really pinpoint how the model is attributing different importance and most importance, so the peaks of this attention, to the seed, so in red, and the bulge regions, which is consistent to what has been previously reported. And second thing, at the same time, if we inspect these attention profiles for the 5p isomiRs, so isomiRs that had modifications at the 5' end, we can essentially see how the most important regions highlighted by attention gets moved, so like kind of shifted, based on the number of nucleotides that were inserted or deleted. So to conclude, we saw in this project that there is a high heterogeneity in the quality of public CLIP-L data, that the transformer networks in general performed better than other less complex architectures, even in our case where we have very short nucleotide sequences. And then about interpretation, that the seed region only is not sufficient for binding, but also other regions are important. And lastly, that the target recognition related to isomiR can be mainly explained by dispositional shifts. So like if you have insertion and deletions, the number of the nucleotides that you modify is really redefining the positioning of the other regions. And to conclude, I would like to thank my co-authors, Huey and Ranjan, all the people in my lab in Munich, and also the university for the finding, and the audience for your attention. And I'm available for questions.
Thank you. Here. Thank you. That was real nice. Right here. So basically what you saw is that, if I understand, is that when it's shifted by 1, let's say, then the importance shifts also by 1. So the original seed kind of is still the important thing, even when it's shifted, right?
Exactly.
Which is very interesting. And I wonder if that's an effect of the model learning, that seed, the seed is important in the more common conformation, like not in the isomiR, but in the original, or is it something, or do isomiRs just target differently? Do you have, if you have any thoughts?
Yeah, so basically the evidence that we have so far, not only for attention maps, but also other kind of interpretation that we tried, is that really the seed is really moving. So what matters is the position of the start sequence, and then that's what the model really captures.
Okay, yeah, thanks for nice talk. So I have a questions regarding your the length of your target sequence. So how do you define this? Because another potential factor that could affect microRNA binding is probably the self-folding of the target sequence. How is this considered for your model? Yeah.
So you're asking how we define the target sequences in
What's the length of it, and how, or did you consider? Yeah, and did you consider that potentially they could have this self-folding that prevents microRNAs to access to the target site? Yeah.
Yeah. So first, so we start from the data that we have, right? So we have the definition of our positive sites, which comes essentially from the experiment. So we have the target length is defined by what we retrieve from the chimera, meaning that in the end we have something like up to 40 nucleotides. And the second thing, the negatives are then basically sampled from the 3' UTR, always with the same length of the positive. And we, so two things. So the positives are heavily filtered to discard any like false interaction that we might have. So we have few steps. Maybe I have something on the appendix. We have few steps that we perform in addition to this bias correction thing, with which we kind of try to ensure that these interactions that we have are actually the most stable ones, of course, by proxy, so running things like RNA co-fold or iRNA. So that we kind of discard everything that we think might be not as stable or really was captured by the clip L, but maybe it's not real.
Okay, thank you.
Thank you very much. We have to move on to our last short talk. So this will be by Cho. Oh, yes.